we are really proud. It's a great honor for me personally to host Professor Paolo Bernardi, since my supervisor, Professor Micheladze, he is, I think, best specialist in Georgia, in mitochondria, maybe not only in Georgia. So my first steps in science was, he explained to me this respiratory chain, and all my students at bachelor level knows that the first question is about the inhibition of respiratory chain and mitochondria. So for me, today's lecture is part of my first steps in science and also the future, since we also want to continue such studies at Ilia State University. I want to ask Professor Margolis to introduce his friend.
Thank you. Yes, so we continue our series of international lectures in different topics related to molecular biology, we had immunology, virology, and today about mitochondria. So what is the common topic of all these lectures? The common topic is that we hear the cutting edge, as they say in America, cutting edge science from the top world-renowned experts, of which Professor Bernardi is definitely one. So Professor Bernardi came from Italy, as you know, from the capital city of Padova. Why Padova is a capital city? If somebody was there, it's not a capital of Europe, Brussels is, it's not capital of Italy, Rome is, but it's the capital of mitochondria science. And it's a world capital of mitochondria science. It's due because Professor Bernardi made this university and this town, the city, a capital of world capital of mitochondria. And that's not only my opinion, because I'm not an expert in mitochondria, my specialty is virology, but recently I have been to a symposium in Padova last year to honor Professor Bernardi, and many greatest experts from the world, different countries came to, and clearly they give credit to Professor Bernardi from major advances in mitochondrial science. So he's definitely number one. When I say he's number one, I don't exaggerate. I know that in Georgia, at the table, as a toast, you want to exaggerate, like saying to everybody, even people who are not so close, that Hashem har perveli magabare, which is Chemi perveli magabare. But in that case, Professor Bernardi is number one in the sense I know one great cello player said about another cello player, the interview he was asked by the journalist, who is named another cello violinist, and said, oh, he is a great violinist, he is number two. And the journalist said, clearly understood that that guy thinks about himself as number one, but just he said, and who is number one? And said, well, there are several number ones. So I'm sure that Professor Bernardi is one of the several, but you can count really on one hand fingers how many real great experts are. So Professor Bernardi was graduated from University of Padua. He has a medical degree, and then he went to MIT, Massachusetts Institute of Technology, one of the several best places in the United States, where under the mentorship of Professor Harry Lodish, one of the greatest scientists, he worked several years and then returned to Italy, unlike many, I must say, people who go to the United States. And many of these talented people who come to their home country, well, they develop something but become the secondary after the top laboratories they worked in the United States. And that was different with Paolo Bernardi, because he came back to Padua, to his alma mater, and made, as I told you, his laboratory and University of Padua really the greatest place in mitochondria. So continue to be on top, competing with all people in the field who are working in Germany, United States, and etc. So I think I'm very proud that today, and you should be proud, that today we listen about great science of mitochondria from the internationally renowned top expert. And I would like to congratulate you, our university and administration, that we managed to present to you science in this high level. Thank you, Paolo, for coming.
Thank you very much. This was too much, of course. You can tell he's one of my oldest friends. But it's good to have friends in science. That's why we do it also. You meet a lot of fine people. I'm grateful for this invitation. I'm very proud to be here. I had a great time yesterday with the students, today with a larger audience. And so thank you, Professor Micheladze. Thank you, Professor Żurawliowa. We share many things, as you will see from the talk and then from the discussion, I hope. It was a nice, warm welcome. I always come back with pleasure to Georgia. It's my 4th time and not the last, I hope. I want to stress that I followed in the footsteps of a giant, my mentor, whose name was Giovanni Feliciazzone. He's the real founder. I'm glad I could continue the tradition by training excellent students who are now established investigators. It's true that I contributed to keep the school, but I don't want to overrate myself. It's good to have good mentors, and I'm impressed by the quality I've met so far in this university, so you should be proud to be part of it, and I wish you all the best. So I have to tell you that today I'm going to cover a general topic, you can tell from the title, and I'm not an expert on evolution. So what I was asked to do is give you some basics of mitochondrial evolution and physiology so that we understand where do we stand and what is the future, hopefully, the future of the field. So I myself am fascinated by mitochondria, their complexity, their beauty, and I pose questions for which sometimes I don't have an answer. And I think that's also the right attitude. We are all students, and we should never take anything for granted. So I'll try to build a story and take you through. This is usually what I use as an introduction to our graduate students when they start the course, and so it's kind of basic. I know that many of you never heard what mitochondria are, several at least. So I'll try to jump from very general to more specific for those of you who in fact know perhaps more than I do. So history is easy because we just see what has been done, how things have been discovered. You don't need to understand much about the science here. So mitochondria were rediscovered in 1890, and I admire the old scientists, how creative they were with means that to us looked like granted, you know, a microscope. It was a major advance, and it was Altman in 1890 to describe bioblasts, he called them. He noticed that these are in all cells, in all cytoplasmic structures, and immediately he saw they were similar to bacteria. And the conclusion he had, just by looking at them, was that they are elementary organisms living in cells and performing vital functions. I want to stress this point, living in cells. He realized they were organelles, something that had life of its own, just based on the morphology. Benda, 1898, was luckier in the sense that he called them mitochondria, and that's the name that stayed with them. And the reason why we call them mitochondria is that they can appear like filaments, mitos, or granules, chondros. This is also something, nowadays there is a lot of discussion about fusion, fission of mitochondria, shape changes. This guy not only saw it, but he called the organelles after this fact. So we keep rediscovering the wheel in a way. So we should not forget And this was there. Great scientists. Altman was right, because now we know that mitochondria carry out respiration. These are the only organelles that are able to use oxygen as an electron acceptor, so it's really unique. And they conserve the energy. They transform the energy of nutrients, what we eat, into chemical energy in the form of ATP. Now, how this is done kept the best minds of the 20th century busy for like 60 years. Major challenge. How do you couple a redox reaction to a chemical reaction synthesizing a phosphorylated compound? And remember, ATP was only discovered in 1932. So, it took a while. Now the key point, which is also in my title, mitochondria most likely, although we know it for sure actually, but most likely is the right word, because in evolution it don't demonstrate anything. It can go backwards. We were not there at the time. We can make deductions. So, most likely derived from ancient bacteria which became endosymbionts with mutual benefit. And we owe this great intuition to Lynn Margulis, 1970. Then it was discovered that they had their own their own DNA, mitochondrial DNA, and they replicate autonomously inside the cell. So Altman was right, because they replicate on their own. This is Lynn Margulis. She should have gotten a Nobel Prize. This was an absolutely revolutionary discovery, proposal she had. Women have a harder time in science. That happens with the discovery of the double helix. To me, she's at that level. I don't care whether she got it or not. She's been inspiring for generations of scientists. And again, this paper is 1914, and they didn't have apparatuses to make high resolution pictures, so they made drawings. And this is what Lewis and Lewis, husband and wife, observed. And you can see some organelle. Maybe you can turn those lights off so we see the slide better. Thank you. You can see the granules, the filaments. This is anatomy, so it was very descriptive. But it's exactly what we see them like these days. They oscillate between granular and filamentous form. Now, don't worry if you're not a specialist. It says late acquisition of mitochondria by a host with chimeric prokaryotic ancestry. It's much simpler than that. The question is, when did this colonization occur? Okay? So, for the non-experts, you have a cell, you know what a cell is, which metabolizes, let's say, glucose. And then at some point in life, this bacterium enters the cell, and the question is, when did that happen? Did that happen before it became a nucleated cell, or after? So there are ways of addressing this by analyzing the proteins that are expressed in organisms that are older than we are. And you can put them in a sequence now by sequencing the genes and then deduce what the proteins are. I'm not an expert. I read these papers because I'm like a child, you know, I find them like magic. So I'll just give you the references, and if you want, you can go see them, but I only tell you the conclusion, not how they got to the conclusion. And there are three theories. One is mitochondria colonized this protocell very early. It didn't have organelles, it didn't have a nucleus, or when they already had endomembranes, or when it was already eukaryotic, meaning it had a nucleus. And by clever comparison of proteins, they came to the conclusion that it's late. So cells existed before mitochondria colonized them, and when this happened, this is very important because it means that mitochondria had to integrate with a nucleus which was already there. And this is extremely important and interesting for what I'm going to tell you next. So for the non-expert, at some point in evolution, these bacteria, protobacteria, and the cell had mutual benefit. Let's see what that benefit will be. First of all, people felt this was impossible to replicate. This happened 400 million years ago, something like this. I may be wrong, nobody really knows when that happened. And so it's a one-in-a-lifetime event. until this paper appeared. It says, inducing novel endosymbiosis by implanting bacteria in fungi. They did mission impossible. They were able to induce Mycetohabitans rhizoxinica, whatever that is, implanting it in a fungus, and over time, this bacterium was transmitted to the progeny, and there was genetic adaptation. So this is the first model ever through which we can study the development of this endosymbiosis. I don't know what this means to mitochondria, but definitely it will lead to understanding how the bacterium and the nucleus talk to one another. And for sure, the cell is alive and it produces proteins derived from the genome of the bacterium. So this is really shocking, and in fact it appeared in Nature, and it's the first time that we can mimic evolution. Do it with Escherichia coli, it doesn't work. It stops there, it's eliminated. So it has to be compatible, and probably this is something similar to what happened during evolution. But now, let's go back to history. This is so cool. As you know, life on Earth was anaerobic initially, there was no oxygen. So the first living things were metabolizing substrates, producing nitrogen, using nitrogen, producing whatever, but not oxygen, until nature developed cyanobacteria. Cyanobacteria have an apparatus to use light for energy, like today's plants and chloroplasts and things. And photosynthesis, as you all know, produces oxygen. But the process was so amazingly efficient that this species took over and they started to develop oxygen at high levels. So atmosphere started to become bluish, not methane, but oxygen. And as you know, oxygen is very reactive, and so it's toxic. And it leads to so-called oxygen catastrophe. Don't think it happened overnight. It took millions of years, but the growing oxygen concentrations were toxic to basically all living species. Lethal effect of oxygen. So anaerobic species disappeared. It was a selection process, and I'm sure in biochemistry they teach you a lot of things. We exploit oxygen because it's so reactive, but we tame it. So we transport oxygen, but we need hemoglobin for that, and that's iron which keeps it there and then releases it. Same thing for mitochondria, as you will see. So this event is known also as the oxygen crisis or great oxidation, and as you see, took place about 2.5 billion years ago. Siderian period, Paleoproterozoic era. I know nothing about this, but long ago. Now, it's at this point, and I'll tell you when it happened. That's another very interesting thing. Not exactly the point in time, but when in the history of evolution of oxygen that occurred. Today we have archaea and bacteria, prokaryotes. For those of you who are not a background in biology, they have no nuclei. And they reproduce by binary fission. And they are fast, because it's 1, 2, 4, 8, 16, and so on. And it doesn't take long, right? And then we have eukaryotes, which have a nucleus. Animals, fungi, and plants. But of course, this comes from a common progenitor. And what happened at some point in the branching, when the nucleus was formed, this is when mitochondria were acquired. So, there is one thing I learned, which I find extremely exciting. It's called One Step Beyond the Ribosome, the ancient anaerobic core. This is an interesting contribution by Bill Martin, because the simple message, I won't tell you the complex one, is mitochondria colonized our progenitors before oxygen appeared. Before. And in fact, it's written there, you can find a common ancestor that had the ATP synthase. I'll tell you about the ATP synthase during the lecture. The same thing we have today. But those protomitochondria did not have a respiratory chain. because there was no oxygen. So this was acquired when oxygen appeared, and you will see how that was such an advantage for us. They did have transport systems for ions, so they could transform ion gradients, which existed, sodium particularly, into ATP. The earliest ATPases were sodium ATPases. Then came the respiration, but we get to that point. The importance of this paper is here. It shows or supposes when the event occurred. This is a genetic tree. We all know how it is done. You can sequence the genome and see how similar species are between themselves and build a tree. And what we know is that there must have been a progenitor common to mitochondria and other species. And it sounds like weird that the closest genome to our mitochondrial DNA is a pathogenic species, a rickettsia, which is not a virus but cannot replicate outside of cells. It's infectious and it causes typhus and anthematicus, which is very serious. And it's interesting because it's already predetermined, it needs to colonize a cell, and look what, mitochondria derived from something which had the same features. So it was a good one, a good guy, but of the same families as of today's rickettsia. This is amazing, and you can find it. Of course, this is not so updated, but it doesn't matter. So what do they look like? Back to the non-experts. This is a sperm cell, and the reason why I'm using a sperm cell is that that's where Altman observed them. And they are wrapped around the neck, next to the head, you see. This is the flagellum. They provide the ATP, so this is swimming very fast. So depending on the cell, this is a muscle cell. These are the dark organelles in between the fibers. This is a higher magnification. Don't they look like bacteria? And this is what suggested to Altman that they were living inside the cells. These are fixed samples, and this is the famous scheme that George Pallady developed. He got a Nobel Prize for this and other things, his observations. Today we know that this is partially an artifact because they are fixed and then contrasted with osmium you have to do that to see them under the electron microscope. The real structure is more dynamic. It's made by these deep invaginations that contact one another. Only a few have been left on this slide, otherwise we wouldn't see much. But just to tell you that today we can see things much closer to what they are in reality by high energy cryo-electron microscopy and other techniques that are changing science these days. But again, let's not forget the old guys. I admire very much Dr. Bereiterhan, who in the 90s was making pictures, movies, of mitochondria fusing or separating. He didn't understand the mechanism, but it's fascinating to see. So this is pictures taken from one of his frames. This is seconds. So you just observe 2.6, 7 seconds, 10 seconds, 15 seconds, 25. In 30 seconds, you see the shape change. Interesting thing, they have fused, and as we'll see, they mix their contents, so they're passing information by physical contact. A lot of vesicular traffic, and I'll tell you something about these two people sitting in the first row, but later. So the opposite, we start with the fused mitochondrion, and you see again 40, 42 and a half, and so on. It ends up first becoming two mitochondria and then becoming another fused cell, fused mitochondrion. Today we know that they can go from individual to network, and the way you do this, you transfect one in the matrix with a red fluorescent protein, the other is green fluorescent protein, and the beauty of this is that it tells you if they're mixing their contents, and they are. When they fuse, they become yellow. So we know they're exchanging what they have inside, and we can separate inner membrane fusion, outer membrane fusion. I didn't mention they have two membranes like bacteria. We know that these are regulated by specific proteins, mitofusin, which promote fusion, and OPA1. My former student Lucas Corano is now the superstar of mitochondria, and I'm proud he's my former student. That's what probably Lonio was referring to. He's much better than I, and that's the beauty. If your students are not better than you, you are not a lucky mentor. So I was lucky enough in that respect. And then you have fission proteins like the RP1, FIS1. So it's a dynamic, and this is the basis of many diseases I will not go into, but it's an interesting part of the physiology. They are social organelles. This is the review that Luca wrote, and I recommend it very highly if you are interested in this part. So mitochondria have a life cycle. They fuse and stay together, and then during the life cycle of that cell, they separate into smaller units. Why is that? Because they use electrons and they undergo degeneration. Some mitochondria become dysfunctional. And if you can single them out, if you fragment them and you have a method to detect the bad ones, you can remove them one by one and leave the good ones intact. So it's a very effective system, this fragmentation, to remove the mitochondria that would do damage instead of do good to you. So that's one of the main reasons why we have this. The second reason why you want to fragment them, what is it? When the cell undergoes mitosis, you want to distribute the mitochondria in the daughter cells. And to do that, they have to be fragmented. If they're not fragmented, the cells cannot proceed in the mitotic process. So there must be a signal, but I will not go into the details. I just want to underline that based on in vitro study, you may think that these processes take minutes because you see them happening, right? In the real world, it's much, much slower. This is a beautiful in vivo study on the turnover of mitochondrial proteins in a mouse. So in general, what is hours in an isolated cell can go to a week, becomes months in the living animal. So don't think this is happening as I'm speaking. It happens, of course, because we have many mitochondria, but not on the time frame that you have seen in those pictures. It's much slower. This is a great study. So, mitochondria, their DNA is different from that in the nucleus. They have small genomes, circular, just like bacteria. And now for the non-experts, you inherit your mitochondria only from your mother. Only from your mother. And there is a deep reason for this, although we don't really understand the mechanism. And I'll tell you that in a second. So when the sperm comes carrying paternal mitochondria, it fuses with the oocyte, and the first effect is dilution. It's a big oocyte, a small sperm, so the parental DNA gets diluted. But even that is not enough. You do not want to pass that on to the next generation because sperm cells do not select bad mitochondrial DNA. They don't have a check-out system. Okay, so since mitochondrial DNA mutates at a faster rate, lethal mutations could be introduced more easily. Females are smarter. The oocyte, as it matures, has a method of selecting the good DNA, even if it allows some mutation, which is essential for evolution. So you have to get a balance. You must allow some mutations, and tell you why. It could be beneficial. But at the same time, you want to prevent the deadly mutations from being transmitted to your progeny. And oocytes have a fantastic check system to remove the most dangerous mitochondrial DNA mutation. Sometimes it fails, but it would be much worse if it had paternal DNA, which does not have this check system. So these mitochondria get ubiquitinated immediately and they get removed. Not one makes it. So we are all clearly, we owe this to our mothers. So in the end, you get maternal DNA and maternal mitochondria, which does not mean that all DNA molecules are the same. There are small variants. That's why you see red and green dots. They're all working fine, but they are not identical. And when the embryo develops, these are distributed randomly So you may have the prevalence of red or green mitochondria. You understand what I'm saying? Slightly different versions. And the key part of it now is that, as you all know, nuclear DNA comes from both parents. So that's how you know, oh, he has the eyes of grandpa, he has the hair of mother. This is the nucleus, of course, our identity. Mitochondrial DNA is more boring, as you will see, encodes only a few proteins. But, as I said, it's inherited only by the maternal lineage. And this is so important because it allowed to establish that we all derive from the same mother. I love to say this at public lectures because it's the basis of an anti-racist attitude. They don't realize, black and white and yellow, we are all derived from the same woman who was born and raised in southern Africa. And this was between 140 and 200,000 years ago. This is a fact. And use the argument, you know. We really are all related. If you go back enough, and I'm talking like 50 or 60 generations. So this is the scheme. How do we know? How do we know? By sequencing mitochondrial DNA. And doing this, you see this? That's our mother Eve, and we all carry her DNA. Now, over time, it's been modified by mutations, and this is the tree of mitochondrial DNA as of today. At the center, you have the most ancient, so you go backwards by sequencing it until you find one which is unique, and then you check how many differences are there, and you make families. So it's done by statistics, if you wish. So they're not infinite, and through this, we could follow human migration. When I get to this point, I become very emotional, because you can really see the history of mankind from our earliest progenitors. L1 was her, it's called haplotype, her sequence. That generated L2, L3, and an M. These are arbitrary names, just tell you how different or similar they are. So we know that first our progenitors crossed across today's Iran, the Arabic Peninsula, and so on. So Europe gave rise to these, and then we know there was a migration. We know that there was land between Europe and the United States, and so on. And then they also traveled by boats, and all the way down to South America. Travel today mixes up genomes much faster, but still you can find a predominance, because these traits do not predispose you to diseases, but they probably are adaptive. And I'll tell you what that means now. This is my great friend Doug Wallace, one of the most important living scientists in mitochondrial science. They gave major contributions to so many things. He was the first to identify a mitochondrial DNA disease, but he was also amongst the great people who did this and demonstrated our origin. He thinks, and there is reason to believe him, that mitochondrial DNA mutates faster so that we can adapt to changing climate conditions. And the determinants are obvious. It's temperature and altitude, oxygen and temperature, because mitochondria use the oxygen, as you will see in a moment, for energy conservation, but also for heat production, which is as important. So his idea is that mitochondrial DNA adapted so that the coupling efficiency is lower in Africa, where you want to dissipate energy in the form of heat, and much tighter in Eskimos, where conserving the heat is more important. I'm just using a simplistic terms, but there is some ground for this. Now I have to decide. I don't want to bore you, but this is the map of the human mitochondrial genome. If you're not familiar with it, don't worry. The point here is that it encodes very few RNAs and proteins. So over evolution, the bacterium passed over most of its genes to the nucleus, and we'll see why. So basically what is left in mitochondria is 11 subunits of the respiratory chain and 2 of the ATP synthase, plus the tRNAs and the ribosomal RNA, which you need in the matrix to synthesize them. What you see here is a scheme of the respiratory chain, and the nuclearly encoded subunits are blue, the mitochondrial are in red, very few. And this is the ATP synthase, only 2 subunits are mitochondrial. Just briefly now, to tell you that this DNA, if mutated, can give rise to a variety of diseases. They are all written here, and I'm not going to read them, but they are syndromes. So the same defect can cause different syndromes, and they are so complicated for a reason which is illustrated here. So they are exceptionally variable, and they can influence the brain, the eye, the hearing, peripheral neurons, muscles, the heart, the liver, kidney, intestine. Why is this so generalized? Because we all have mitochondria everywhere, and these diseases develop over time. Some are early, some are later, and this is due to the fact that over cell division, after cell division, the sick mitochondrial DNA could become predominant. But I don't want to discuss this part. I just want to tell you that mitochondrial diseases in the proper sense are those caused by mitochondrial DNA mutations, and these are inherited by the mother only. But the vast majority are classical Mendelian diseases because most of the genes are in the nucleus anyway. Okay, so let's not mix up and think that all mitochondrial DNA, all mitochondrial diseases are due to mitochondrial DNA. It's just the other way around. Mitochondria have 1100 proteins, more or less, and only 11 or 13 are encoded by their DNA, so the vast majority of diseases comes from the nucleus. But then there is an emerging third category, which is diseases with a mitochondrial pathogenesis. I gave you an example yesterday, when good mitochondria are still part of the death circuit, and at the end, if you have patience, I will show you briefly how is that done. Now, this is going to be for the specialist, but it's not going to take me more than 2 minutes. But it's a major question. Why do we still have a maternally inherited mitochondrial DNA? Why did mitochondria decide to transfer 1100 genes to the nucleus and retain only 13? Why is this? This is a big mystery of evolution. And Doug Wallace dedicated this beautiful review. So I leave you with the review, and I'll just go briefly through the hypotheses. So if you are not an expert, you can sleep for 1 minute. So the idea is that these are, not by chance, related to the respiratory chain and ATP synthase. This is for me to remember what I have to say. As I said, mitochondrial DNA mutates faster. What is faster? It's 2.5 substitutions per site per billion years. So don't think it will happen overnight in your mitochondrial DNA. It's a very slow process. But still, this is 20 times faster than the nucleus. The nucleus is more protected. It has better ways of compensating mutations. So nuclear DNA mutates slower, mitochondrial DNA mutates faster. So in principle, it can provide a better fit. But if it's a big genome, the chances of bad mutation is higher. It's called the Muller's ratchet. This is why, over time, the mitochondrial DNA became smaller and smaller and smaller and smaller. It's an advantage because it replicates faster. It's a circle. So if it's a small circle, it's faster. And so this was an advantage to select smaller mitochondrial DNA. And this way, of course, it's complicated because proteins have to be synthesized by mRNA and imported into mitochondria, but this is what actually happened. So, why did they decide to keep these genes? One is simple. Over time, the code in mitochondria, genetic code, differed from the code of the nucleus. So, the phenylalanine triplet is a stop codon in mitochondria. So they made it to be sure that proteins that were not meant to go to mitochondria were synthesized on mitoribosomes. So it happened for some reason. So now we can no longer do this, because if our mitochondria decided to give up a gene, this could not be interpreted by the nucleus. Is that a good reason? Maybe not. The other three are related to how far should the protein go to be inserted and imported in mitochondria. And basically the next three hypotheses, I will go very quickly, say that the most hydrophobic proteins were retained, those that need to be assembled immediately in the membrane without being translocated. And in fact, all the core protein of respiratory chain are mitochondria. So I'll go quickly because otherwise it will be too long. Just one small experiment. Somebody had the idea, what if I take subunit A, which is mitochondrial, and what I do, I create a synthetic gene which is now interpreted by the nucleus. It encodes the same protein, and I put a mitochondrial targeting sequence, and I ask the question, will it be functional? I shut off the mitochondrial form and I force the nucleus to make subunit A. Will that work? Yes. It worked. So in principle, we could still export mitochondrial sequences to the nuclear DNA. It was done in mammals, it was done in yeast, because in yeast subunit C is encoded by mitochondria. It was forced to go to the nucleus, it worked as well. So probably we can't do it anymore because of the genetic code variation, but the real reason is we don't know. Nature beats us. We don't really know because some early eukaryotes still have the same genetic code and still maintain the same proteins that we did in mitochondria. So we don't have a complete explanation, but this is the beauty of science. So now quickly, as I said, we may have normal mitochondria and mutant mitochondria, and mutant can be good or bad. In this example, it's bad. And what happens is that during mitosis, they may end up in the same cell, and you need enough bad mitochondria for disease. It's called the threshold effect. This is what makes it different from Mendelian inheritance. So some cells can be healthy, some cells can be sick. And we have identical twins, some are sick, and the brother or sister is healthy. So this is really bad chance if you get enough of the bad mitochondria. There's been an experiment, and this has been authorized in the UK for some time. Then you can cure the mitochondrial DNA disease by enucleating the oocyte. Okay, so now you have an egg with healthy mitochondria, and you take the nucleus for the donor patients, the woman affected by the disease, and you can fertilize this new egg, and it will have all the features of the parents, but not the mitochondrial defect. It's a risky practice. It does allow growth of normal babies, but if you do the experiment in mice and you create these chimeras that have mitochondria from one donor and the nucleus from the other, turns out that over time they develop diabetes, they become obese. So these mitochondria are adapted to this nucleus, and you can't tamper with that. And it's co-evolution. So not all mitochondria are the same, and we don't understand why, because they perform the same function. So maybe Doug Wallace is right. You can't give an African person the mitochondria of an Eskimo, because it's going to predispose him or her to disease. That's the idea. So it's really hard to eradicate these types of disease. I should go quicker, but the question is, why do we have mitochondria? And of course, you need to know this. If you utilize glucose without oxygen, you can make 2 ATP molecules per glucose. ATP is our chemical energy. I can't move, I can't think without ATP. If you have mitochondria, you can oxidize completely the glucose to CO2 in a Krebs cycle, and when you reoxidize the reduced cofactors, you make 32 ATP molecules. This was the big jump. Without this energy yield, we could not move. Only This allowed the formation of multicellular organisms for the simple reason that otherwise you would not have enough energy for movement. And to get your prey, you have to move. No mitochondria, no animal life, no plant life on Earth. How do they work? You are all familiar with the hydroelectric dam, where you have a reservoir of water. Your potential energy is the jump from here to the penstock. This drives a turbine. This is the outlet. Then you have a generator, a powerhouse, and then you transform the electricity into long distance by playing with the voltage. Mitochondria do exactly this. So the reservoir is reduced substrates, plenty of electrons. They want to go to oxygen. And they go to oxygen down a gradient, which is exactly like a hydroelectric dam, and reduce oxygen to water. The energy is not wasted because we have mitochondria. Mitochondria have electron carriers that at the same time are proton pumps, and they generate a proton gradient, which is used to make ATP and export it to the cytoplasm. This is exactly what happens. You need something transforming electrical energy into proton gradients, and the protons become now power sources for ATP synthesis. We owe this to the genius of Peter Mitchell, single winner of Nobel Prize in 1978. I will skip this. This is your ATP coming out of the cell. Then the structure of the ATP synthase was established. This is not a cartoon, this is real. Each of those little spaghettis is a protein, and that's the way it looks in the real world. There is a real world of microscopy, which we can do at high resolution now. It's made by a catalytic part, and they snap into one another to make like a V engine. How does this work? I have a movie. You want to see my movie? Let's see, it was working before. I need to find... Bravo, yeah. But then we have to go back to yeah, it's now bravissima.
ATP synthase is a molecular... ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP.
What you see is happening for real. The little dots are the protons.
The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy.
Chemical energy made...
The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP.
Now you will see the real thing, the single proteins that make the head.
The F1 ATPase portion of ATP synthase has been crystallized.
The music is not so good. Now we'll see what happens within the catalytic portion. You see the rotation is mechanical energy.
Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft. Like any enzyme, ATP synthase can work in either direction. If the concentration of ATP is high and the proton gradient low, ATP synthase will run in reverse, hydrolyzing ATP as it pumps protons across the membrane. To show the rotation of the central shaft, a short fluorescent actin filament was experimentally attached to it. Single filaments attached to single F1 ATPases can be visualized in the microscope. When ATP is added, the filament starts spinning, directly demonstrating the mechanical properties of this remarkable molecular machine.
So, it's very simple. Electrochemical potential becomes a proton gradient, and the proton gradient is used to drive a rotor, and now mechanical energy is used to synthesize ATP just by making ADP and phosphate close enough that the reaction takes place spontaneously. This thing rotates at 130 revolutions per second. Your car will not stand this, 7,800 per minute. Do you know how many of these we have? Well, we're calculating with Dr. Michelazzi and Margolis. We have 10 to the 15 cells, 10 trillions. We have, let's say, 1,000 mitochondria per cell, 10 to the 18. And let's say 1,000 mitochondria ATP synthase is probably more, to the 18th. Do you know how much ATP you make per day? Have an idea? Your weight. So if you weigh 70 kilos, you make 70 kilos of ATP every day. You don't realize it. It's made and hydrolyzed, made and hydrolyzed, made and hydrolyzed. And we calculated, if you buy it by bulk, you can go down to $40 per gram. It means $2,800 per day you cost. It's truly amazing. It gives you an idea of the amount of energy it takes to survive. That's why we need calories. That's why we need to eat calories. And this is what mitochondria do for us. These spinning machines, they hum, you don't hear them. I find this, even my granddaughter is fascinated by these things. So ATP is made, but another key in that experiment and in what you've seen in the movie is that when they don't have oxygen or substrates, mitochondria become ATP consumers. I think I will stop here because I have more, but it's getting too long. We're giving you a message, which I find my students love this. So remember that in the end, mitochondria derived from parasites which wanted their own good. So why did they stay with the cell? And why did they find this compromise? The cell provides constant temperature, I mean our cells in mammals, let's say. Temperature, substrates, and oxygen. So they are quiet and they become very efficient. It's like pushing a small gear in your bicycle. And we push a small gear because we go faster this way. That's how it works. So they adapted very well. And the trade-off for the cell was maximizing the amount of ATP that could be made, because on their own they could only make 2 molecules, right? So it's a good trade-off. But the moment you get hypoxia, mitochondria stop being your friends and they steal the ATP from glycolysis to maintain their own gradient. They become, again, the selfish organelles they started with. The analogy is, I see a lot of smiling faces from young people, but also smiling faces from older people. But the reason why I talk about the young people is that you have a lot of friends, you are popular, you go to meetings and everybody wants you, and you're well liked, until you break your leg. Oh, Jesus. Well, nowadays you heal fast. Let's say you're sick, you're at home, some people start calling you, a few show up, you're sick for a month, very few show up, you're sick for 6 months, only your mother comes close. This is like mitochondria, good friends and bad friends, you know, it depends on the circumstances. how you define them. Anyhow, the very last thing I wanted to say is that we live in a difficult environment because oxygen is still around us. And remember, mitochondria are like cables carrying electrons. Do that in water, you die, you get electric shock, right? And this is similar. It's insulated by the membrane, but some leaks of electrons occur all the time. It's called reactive oxygen species, which is part of pathology. I had these slides from my friend Anatoly Starkov, but I'll go very quickly. All these little errors that you see are electrons that are leaking out and reducing oxygen 1 electron at a time. It's useful because we need this, for instance, to make for monoamino oxidases. They produce ROS, but it's important to create mediators. But the point is that once again nature beats us because it uses low levels of ROS for signaling, while high levels of ROS are part of cell death. So again, the dual nature of mitochondria, but the beauty of nature, which exploits all means to ameliorate our responses. Final, this is now very easy, so relax and sit back. Is this over? Do we know everything about mitochondria? Absolutely not. What you see here is the fraction of papers published on mitochondria, and it's this little line here. This is the nucleus. What happened when PCR appeared, all got a boost, but when mitochondria overtook the nucleus, was the discovery of apoptotic signaling by cytochrome C, which is released when mitochondria participate in apoptosis. These two papers, which we all took, Zeldon Wang, who's a good friend, a great scientist, think about this, 16, and this is a few days ago, now it's even higher. It's a huge number of citations. This really changed the field for all of us, and the reason is, I mentioned mitochondria, I mentioned ROS, reactive oxygen species. Because of their role in cell death, there is new life to our field. And actually, because of these pathways to cell death, mitochondria are involved in cancer, neurodegeneration, inflammation, senescence, basically everything. They are at the heart of today's medicine. And this is because signaling is going on. I mentioned Elena Juravlyova and David Micheladze, because they have done important work on signaling, and this is what the one I like the most, is sigma-1 receptors, which are involved in those fusion-fission events, not only between mitochondria, but with other organelles as well. This is only a fraction of what they do, but it's nice to have good colleagues in Georgia, and I'm really fortunate that they are in the audience today. Okay, so this is a summary that you can read in this review, how many things have to do with mitochondria, metabolism, ion homeostasis, interactions, heat production, gases and ROS, DNA extrusion, steroid hormone synthesis, ATP production. So it's a long life to mitochondria, from death to life. Thank you for listening. It was a bit long, but what can I do?
Thank you. Questions?
Don't ask too difficult questions. There are no stupid questions, but there are difficult questions for me to answer.
Maybe first question. So mitochondria could be the target for cancer therapy. And what about if mitochondria can steal the ATP from the cancer cell? How can we use it to treat the cancer?
Yeah, it's a clever observation. The problem is always selectivity. We know that mitochondria have to do with cancer, in particular when they don't want to activate the apoptotic pathway. So the classic is BCL2 proteins and like venetoclax, these drugs that force BCL2, and then the selectivity comes from the fact that you only activate the death pathway in the transformed cells. But that's one example which is the opposite of what you asked me. The idea is, can't I target mitochondria to become dysfunctional, but only in cancer cells? It's a great idea, but the problem is, how do you get the effect selectively in cancer cells? We have an experimental not trial yet, but we have a system which we have patented. It's mostly my colleague Andrea Razola, but it's exactly your idea. You know what it is? Hexokinase, I don't know if you're familiar with hexokinase. Is the first enzyme of glycolysis, and hexokinase type 2 likes to attach to the surface of mitochondria, because then formation of glucose 6-phosphate is faster, because ATP comes off, the enzyme is there, and you start glucose metabolism. What we found is that by using a peptide derived from HIV, fusion peptide, targeting this peptide to hexokinase, we can force cell death only in cells that overexpress hexokinase 2, that is, solid tumors. So the selectivity comes from the location of hexokinase 2. The selectivity of the peptide depends on the fact that it's activated by metalloproteinases. It's a hairpin, it only releases the active part after cleavage by metalloproteinases, which are in the environment of the tumor. So in animals, this works. So you can direct an activator of cell death. This is calcium dependent, it's through the transition pore, and it's amazingly effective, okay? And that's exactly what you're saying. So in this case, we exploit hexokinase 2 overexpression to target solid tumors. And it's exactly what Elena said. So there is no limit to what you can do as long as you can hit the tumor cell and save the healthy cell, which is a real problem. But that's just one example. There are many, even the toxicants you are studying, if you could direct them to tumor cells, fusion, or through receptors, maybe the signal receptors, I don't know. But that's exactly why mitochondria are important in cancer biology. Yeah.
Another question that's maybe not very real, but you have explained that we can put some mitochondrial gene to the nuclear genome, but maybe we can use also it's vice versa, so put some mutant gene in the mitochondrial DNA and then insert in the cells. Or
it's a good project. It's very I don't think it's ever been tried, actually, but why not? The question is choose carefully, because you should why not? So reverse transfer genes. The thing is, in evolution, it only went one way. It only went one way. Because I am not aware of, but of course you're right, because so many genes that encode for mitochondrial proteins are nuclear. No, I don't think the experiment has ever been done.
Maybe we will have the problems with the out transportation, so teams and toms.
No, but it's a good point. How about the other way around? Yeah, somebody should do it. I think it's doable. We have a student.
Yeah, so you can try. Student first.
Student first.
Can I say that they came into a eukaryotic cell for survival, but still they don't have a perfect environment to survive?
Okay. Evolution always selects the best, and evolution means until you are 16, 17, when you generate a new individual. So in natural, I'm talking about very young people. So probably at that age you don't have heart infarction, you don't have strokes. So it's perfectly fit for reproduction, right? What happens after that is nobody's business. It's just degeneration and whatever. So when the environment becomes bad, there's not much you can do. It's a consequence of the conditions that were enacted when it was good. So I don't think that's a matter of selection. But you are right that in the long term, as life becomes longer and longer, we see the parasitic nature of mitochondria, quotes, under negative circumstances. Because it's a fact that if the proton gradient lowers, ATP from glycolysis, which never stops, is used by mitochondria. They become a death factor. They precipitate the energetic crisis. That's why it's important to remove the bad mitochondria through mitophagy And that's why it's so important to fragment them so that you can pull out the bad ones and leave the good ones. So it's all connected, but it's true. It's still a struggle between two different genomes, which integrated beautifully well, but perfectly is a tall order. Maybe we are not there yet. Evolution is going on. There's no question. Right now, I like to tell patients with rare diseases something that for some reason cheers them up, honestly. Even if you get cancer, there is something you have to think. It's an experiment of nature that went the wrong direction. But it is these experiments that allow us to maximize our health, our abilities, our brain. So in a way, we should accept the fact that mutation is not always bad, as long as we have selection processes that select for the good ones. But since it's random, we have to accept the risk. That's how I like to see it. So in the end, it's still an experiment that nature is making. And really, this is still going on. We're adapting, and God knows which way we will go, depending on the environment we leave to our children and to our grandchildren. So.
And one more question. Are there only one type of bacteria that enter into our body, or it could be more types of cyanobacteria?
Oh, that's a very clever point. So somebody has calculated how rare this event, or how frequent this event is, by using this retrofitted genetics I just mentioned. The conclusion is it's extremely rare, but not impossible. And probably it happened only once in the evolutionary tree, and it's unlikely to happen again. But, you know, with this new model, which I showed you on purpose, it's been done in vitro, maybe we'll discover things, and maybe it's more common than we figured. We have a great virologist here. Another example I can give you is the integration of viral genomes into cells, and whether viruses derive from cells or whether oncogenes derive from viruses. It's both ways. Exchange of genetic material has been known since the 70s, I would say, because before that it was thought impossible that RNA viruses would integrate. But maybe you can answer the question. That's a great example of genetic exchanges, including entire genes between viruses, which are obligatory intracellular parasites, just like mitochondria, and the nucleus. So I think of something similar going on, but they have to be compatible, because this is a whole organelle that has to survive in an initially hostile environment. I just think, what happens at the beginning? Maybe what happened at the beginning is what's happening in that Nature paper. And they are following it up now to see how it evolves. That's the beauty of that experiment. But it has to be compatible. That bacterium with that fungus. If they use E. coli, it doesn't work. But now we can do crosses based on that. And you can figure out, oh, this is compatible, this is incompatible, let's make it better.
Thank you.
Well, thank you. We are a very, very creative student. And you know, I'm not an expert, so I can say anything. I don't need to. These things, you can speculate, you know.
We imagine for science.
So when you said that mitochondria became an organelle of the cell because it had good conditions, during cancer, can we give the mitochondria better conditions to maybe migrate to healthy cells?
He's touching upon a very modern thing, which is mitochondrial transfer. It occurs between inflammatory cells and circumstance, and I'm not an expert there. It's an emerging field, and yes, you could boost immune cells by providing them with good mitochondria, or you can target mitochondria to prevent cancer from spreading. The real answer is it's not yet an explored field, but this issue of mitochondrial transfer is a big, big thing nowadays. Maybe it's also a way of evolving mitochondria by lateral transfer, because in the end that's what it is. Thing is, these are mitochondria from the same organism. So one day people will start doing this with transplants, healthy macrophages from somebody who has a different genome and so on and so forth. That will make life complicated. But this is a very interesting question, and there are studies on this, actually, yes. But, yeah, good point.
Okay, thank you.
Thank you. Finally, Dr. Margolis, ask me a question.
No, it's not finally. But,
No, finally means you
I'm
have to
sure
wait your
there are
turn.
more questions. No, but my question is a question of my ignorance. So you mentioned that in evolution, mitochondria, using current terminology, outsourced some of their proteins so that DNA, nuclear DNA, supplies them with the necessary protein.
Yes.
But does it in evolution, cells use this protein for something different from feeding mitochondria? Does this protein have any function outside of mitochondria?
Some proteins have dual functions, definitely, but I'm not sure I'm getting your point on. Yeah?
So you mentioned that some of the proteins needed for mitochondria
most of them, yes,
are encoded
yes.
in cell nucleus.
Yes.
So in that sense, my question is, these proteins, which in evolution were transferred from a mitochondrial genome to a cellular genome, do they have any function not related to mitochondria?
This is a tricky question, because to transfer the genes to the nucleus, they also needed to add a mitochondrial targeting sequence, otherwise it would not be imported. So it has been a very complicated and long process by trial and error. The mitochondrial targeting sequence must have been pre-existing somewhere in the nucleus, and when it happened by chance that they were combined, now it started to be efficient, and now probably mitochondria stopped to make their own. That's how I picture it in my own mind. There's another question. To import the proteins, you need specific import proteins called TOMs and TIMs, outer membrane, inner membrane transport proteins. So it's a chicken and egg question. They could not import proteins unless they already had the machinery to import them, and this is probably parallel evolution. So they developed the TIMs and TOMs before they could import the protein, and that's by mutation, and still these are all genomic genes. These don't come from the mitochondria. It's a miracle of evolution, Vanya. I think it's by trial and error, you try and try over billions of years, you end up with the right combination that has an advantage and moves the thing forward. So it's a very good point. An answer, I don't have an answer. Who am I to tell? It's not even my field, but it's good. We can speculate and ask the real experts. But yeah, dual functions proteins are there all the time.
Just one additional question about the sorting and how oocyte identify the father mitochondria. They do some mistakes, since I saw some publication about few father mitochondria.
This is a great question. No mistakes are made. They get ubiquitinated, and the question is, what is the recognition system? Honestly, I don't know what it is, but it's a fact that undergo selective degradation. There was a paper, by the way, an Italian group, claiming that they could show transmission of paternal DNA. It was published in Cell, like 30 years ago. A recent paper by Patrick Chinnery in Nature demonstrated that those were pseudogenes. So we have nuclear DNA sequences that resemble mitochondrial DNA sequences, but are there independently, and they are transmitted as nuclear genes, so they have Mendelian inheritance. And that's probably the mistake they made, because everything was done by PCR. You can't see the protein. So they amplified sequences in the cell that derived from the nucleus that look like paternal sequences, but they were transmitted by nuclear DNA. They were not because of... It was assumed they were mitochondrial. They were not. It's a nice... Patrick Chinnery, Nature 2 years ago maybe, and he gave a seminar, and it was immediately obvious that probably that explains. So there was no cheating in the STEP paper. They were just amplifying something, thinking it was mitochondria.
So just
it was not.
problem with the design protocol. So they
Yes,
should
but
purify it, maybe.
You do what you can do. At the time, it seemed unexpected but real. Then nobody could reproduce it. It lingered for a long time, and to me, this was the answer. They amplified the wrong thing. It looked like mitochondrial DNA. It was not.
Maybe this process could be the one of the mechanisms how gene from the mitochondrial DNA can transfer to the genome.
Maybe. Sure. This happened. Oh, these are unsuccessful attempts because those are not transcribed. You're right. This thing went on, and some worked and some didn't work. And I think if you do the genomics, you will find this now that you can do it with any sequences. But absolutely, most of the DNA is not transcribed anyway. God knows how many sequences are there that derive from this slow and perilous transfer of genetic material. But yeah, it's really, science is beautiful. There is so much to do. And every generation starts it over. Yeah.
Any other question about the mitochondria? Mitochondria is the main source of life and energy, so I think they spend a lot of ATP.
Yeah, me too.
Due to the enhanced synaptogenesis.
That's a long story.
So,
yeah.
Thank you, Professor Bernardi.
No, thank you for coming. Thank you very much.
I'm simply present from the university, and since we spoke about the mitochondria, so this is the red wine. Red wine contains resveratrol. Resveratrol is one of the most potent stimulator of the sirtuin-3 and mitochondrial bioenergetics. So please do not drink too much, but 1 or 2 glasses, it's enough to stimulate mitochondria.
I like wine, and I admire Georgia as the cradle of wine making in ancient times and the traditions that are still alive. I've been to Kakheti, I've been to many places. Yeah.
Yeah.
Thank you. It's very appreciated. Thank you very much.
Thank you.